Almost every publication by a scientist appearing in the top journals, Cell, Nature, Science, has antibody research in it. Antibodies are also 50% of newly approved drugs. They're incredibly important to make people better when they're sick. There is a horrible problem of irreproducibility in science and a lot of irreproducibility in antibodies, which stems from poorly characterized antibodies that are provided commercially. And scientists often aren't in the business of making antibodies anymore. They leave that to companies to do, and that's a mistake. Scientists should make the antibodies.
IPI is interested in generating antibodies. These are affinity reagents, reagents that recognize and specifically stick, if you will, to unique proteins in the body. And these reagents are incredibly valuable for a number of reasons. One, they're tool compounds. They provide us the ability to actually study the basic mechanism, the biological function of hundreds, if not thousands, of proteins in the human body. More importantly, these antibodies can serve as therapeutics. That is, we can use these antibodies to modulate biological processes in the body to enhance or reduce the immune response to treat a wide range of diseases, including cancers, infectious diseases, and autoimmune diseases. So really, in some sense, the biomedical application of antibodies, as well as the fundamental basic biological insight, is almost limitless.
Various studies have looked at the amount of money that's wasted on bad antibodies per year, and in the US it's about $1 billion. If you've ever worked with antibodies, you'll know the problems you might have. You think it does what it says on the tube, but it doesn't. Proteins are different from, say, buying a chemical or something, and quite often they're badly validated or even badly made. So we're really striving to make the correct protein and raise antibodies as close to wild-type protein as we possibly can to try and make better reagents and better proteins.
We've put together a unique infrastructure here where we've got an enormous amount of automation and robotics that enables high throughput both protein production and the ability to use those proteins to select antibodies.
Key strength of the IPI is tackling this problem of antibody discovery across the full proteome so that we can really make those key fundamental biological discoveries that can then be translated into therapeutics down the road.
The dream of every biomedical scientist or clinician is to make a new drug, but very few people realize that goal. So we want to help them do that. We want to help them do that, and we want to, the diseases they're interested in, we want to help them cure them and make patients better, prevent people from dying, allow them to recover from cancer, get rid of their autoimmune disease, and, have full lives, be healthy.